Here is a combined summary of the 2 pages. Graph Fourier MMD, GFMMD, is a novel distance between distributions and signals on graphs, which is defined via an optimal witness function that is both smooth on the graph and maximizes the difference in expectation between the pair of distributions on the graph. This method is particularly useful for comparing signals on graphs, such as protein interaction networks or single-cell data, where traditional methods for computing distances between probability distributions in Euclidean space are not applicable. The authors begin by introducing the concept of integral probability metrics, IPMs, which are distances between probability distributions characterized by a witness function that maximizes the discrepancy between distributions in expectation. They then define GFMMD as a specific type of IPM that borrows notions from optimal transport but does not require a distance metric, making it applicable to any undirected graph with non-negative affinities. The authors also discuss the Wasserstein distance, which is another type of IPM that measures the least amount of work required to move mass from one distribution to another. They provide a dual formulation of the Wasserstein distance in terms of smooth functions, which is used to motivate the concept of smooth functions in abstract spaces. The graph Laplacian and effective resistances are also introduced as important concepts in graph theory. Effective resistances provide valuable information about a graph, such as commute times, and are used to generalize the concept of commute time to arbitrary probability distributions on graphs. The authors then discuss related work, including spectral distances between graph signals and diffusion EMD, which involves diffusion graph signals to different scales to create multi-scale density estimates of the data. They also mention other methods for computing EMD between distributions defined on a distance graph. Finally, the authors introduce their method, Graph Fourier MMD, which defines a distance between signals on arbitrary graphs that incorporates graph structure. They demonstrate its effectiveness on toy datasets and single-cell data, where it is used to identify gene modules and measure gene localization. The research discusses Graph Fourier MMD, GFMMD, a novel distance measure between probability distributions on a graph. It's defined as the maximum mean discrepancy between two distributions, with the feature map being the inverse square root of the Laplacian. This distance is particularly useful for understanding signal localization and concentration on graphs. The authors provide several theorems and lemmas, including a representation in terms of an explicit feature map, which makes computation efficient. They also show connections to f-spectral distances, resistive embeddings, and expected effective resistance, offering insights into signal properties and graph structure. Computational complexity is discussed, with an O-NLOGN algorithm for sparse graphs. Experimental results on identifying distributions on the Swiss roll demonstrate the effectiveness of GFMMD in capturing geometric and topological properties of signals on graphs. This research paper introduces Graph Fourier MMD, a novel framework for measuring distances between signals on graphs and generating embeddings that preserve these distances. The framework is shown to outperform existing methods in terms of accuracy and speed, particularly in high-dimensional datasets such as single-cell RNA-sequencing data. The authors evaluate the performance of Graph Fourier MMD on a Swiss roll manifold, comparing it to other methods such as diffusion EMD, Synchrn, and RBF kernel MMD. The results, presented in Table I, demonstrate that Graph Fourier MMD achieves the highest Spearman-Rho correlation with the ground truth manifold distances, while also being significantly faster than other methods. The authors also apply Graph Fourier MMD to single-cell RNA sequencing data, leveraging a publicly available dataset of CD8-positive T cells. By computing GFMMD between genes, the authors identify clusters of genes that exhibit localized expression on the cellular manifold. These clusters are shown to be biologically significant, with enrichment for signatures of naive T-cell activation and effector CD8 T-cell activity. The paper also introduces the concept of localized genes, which are genes that have high expression in specific regions of the cellular manifold. The authors propose a novel type of analysis that involves searching for localized signals, which can be used to characterize salient cellular subtypes. They demonstrate the effectiveness of this approach by visualizing the localization scores of genes and comparing them to housekeeping genes. The paper concludes by highlighting the advantages of Graph Fourier MMD, including its rapidity, ability to capture global properties of the underlying data manifold, and its applicability to high-dimensional datasets. The authors also propose future directions for research, including the development of new methods for analyzing localized signals and applying Graph Fourier MMD to other types of biological data. Here is a combined summary of pages 7 and 8 of the research paper. The appendix provides proofs for stated results, including Theorem 4, which bounds the Graph Fourier MMD, GFMMD, between two distributions P and Q in terms of their expected resistances and couplings. The proof involves applying the bias-variance decomposition and Fubini's theorem to establish the upper bound. Corollary 4.1 provides a more specific bound for the case where P and Q agree on a set of size A, with the union of their supports being S. The paper also presents an additional result, Theorem 5, which shows that applying the GFMMD feature map to a family of Dirac delta functions centered at each vertex of a graph G results in a k-dimensional embedding that coincides with Hall's spectral graph drawing. This is proven by showing that the best k-dimensional embedding of the data matrix of Kronecker deltas is equivalent to principal component analysis, PCA, of the graph Laplacian. The appendix also includes additional figures and experiments, including the Swiss roll experiment, which demonstrates the ability of GFMMD to capture nonlinear directions in a linear space in the presence of strong noise. The experiment shows that GFMMD has a clear linear correlation between distances, whereas other methods such as EMD and kernel MMD failed to discern between higher and higher distances. The single-cell localization experiment visualizes the spread of the most localized signals over the graph, using PHATE to produce two-dimensional embeddings of cells in Euclidean space. The paper also presents additional toy experiments, including a grid graph and a bunny graph, which verify that the measure of spread decreases as the signal is diffused. Finally, the appendix includes examples of bimodal signals and localization on the Minnesota graph, which provide further sanity checks for the measure of closeness to the uniform. The examples involve diffusing signals across the graph and measuring the distance to the uniform, demonstrating the effectiveness of the proposed method. The research paper presents a comprehensive analysis of the diffusion process in a system, focusing on the evolution of the signal delta 0 over time. This is visually represented in Figure 8, which shows the diffusions at times 20, 24, 28, and 212. The corresponding distances to the uniform distribution are provided above each visualization. The paper highlights the importance of understanding the temporal progression of the signal as it diffuses through the system. By examining the signal at different time points, researchers can gain insights into how the system evolves and how the signal changes its characteristics. The visualization in Figure 8 demonstrates that the signal undergoes significant transformations as it progresses. At time 20, the signal appears to be highly concentrated around specific points, indicating a strong non-uniformity in the system. As time advances, the signal begins to spread out and become more evenly distributed across the space, reaching a near-uniform state by time 212. This transformation suggests that the system undergoes a process of homogenization, where the initial non-uniform signal becomes increasingly uniform over time. This could be indicative of the system's ability to self-regulate and achieve equilibrium. The paper also discusses the mathematical models used to describe this diffusion process. These models involve complex equations that capture the dynamics of the signal as it evolves over time. The authors emphasize the importance of these models in predicting the behavior of the system and identifying key parameters that influence the rate and nature of the diffusion. Furthermore, the paper touches upon the limitations of the current study, such as the assumption of a linear system and the lack of consideration for external perturbations. The authors acknowledge these limitations and propose potential avenues for future research, including the exploration of nonlinear systems and the incorporation of external factors. In conclusion, the paper provides a detailed analysis of the diffusion process in a system, utilizing both visual and mathematical tools to understand the temporal evolution of the signal delta 0. The findings suggest that the system undergoes a process of homogenization, achieving a near uniform state over time. However, the study also highlights areas for improvement, including the need to explore nonlinear systems and account for external perturbations.